And thanks for the invitation from ProImmune to speak here today. I think what's really interesting is I looked online and I spoke for ProImmune 8 years ago in San Francisco. And I had just finished my PhD, and so obviously my journey has been much different, or it's different now than it was then, and I'm sure it's the same for ProImmune as well. So I'm really happy to speak here today. Also, I feel like I'm not doing Jeremy and ProImmune justice by running in and out, but I do have to run in and out of the meeting, so if you can catch me after my talk, or I'm pretty active in social media, shoot me a message or an email. All right, so let's get started. I can use the keys.
That's fine. I think the dongle will take it out. Should work.
Perfect. I do have to disclose that I'm a co-founder and serve on the board of directors of Monotherapeutics. It should be unrelated to our talk. So as I mentioned, in 2013, we moved from Houston, Texas to DC, where we formed the Program for Cell Enhancement and Technologies for Immunotherapy, or SETI, not the Search for Extraterrestrial Intelligence. And when we started, we were about 3 people total in the cell therapy lab. That includes stem cell processing and GMP processing, and there was about 10 of us doing basic research. We submitted our first IND in September 2013. And for those of you who have never submitted an IND to the FDA, at least an academic IND, so now industry-sponsored INDs have to be submitted electronically, but this is what an academic IND looks like. This is thousands of pages, and you have to submit it in triplicate to the FDA. So we submitted that in September 2013, and on May 19th, 2014, we treated our first patient, well before a year since we had been there. So now we manufacture over 150 products per year. That includes stem cell processing and good manufacturing processing for INDs, and we support about 15 different clinical trials on 10 different INDs. We have about 150 infusions per year. There's about 15 staff in the cell therapy lab, but that's now closer to 20 to 25. And in total, SETI has about 40 different staff members. Our center director, or our SETI director is now our cancer center director, Kath Bollard, and it's been quite the journey for us to take over the last 6-7 years. And before I want to go any further, I do want to thank not only the cell therapy lab, which many of whom are depicted here, but also all the ancillary labs that support us. And that was really the first takeaway I had once I moved to Children's, was how much help we needed from everyone else in order to be successful. And that includes the blood bank, where we irradiate our cells, lab medicine, who helps us with flow cytometry sometimes, the good clinical practice team, who does all our regulatory work, the CTSI, where we infuse the patients, our bone marrow transplant team, where we typically get the patients and who consult with us, and even our environmental services team. I'm fortunate enough to come to Boston to speak with you guys today, but nobody's ever recognizing the environmental services team, who make sure that our facility is clean enough to treat patients every day. So I'll start by talking about bone marrow transplant and how that's a great model for cellular therapies. So bone marrow transplant, because of the necessary myeloablative conditioning, is associated with a number of complications. One of those complications are viral infections. The mortality rate for viral infections is around 39% in a study that came out of Baylor a number of years ago. So what we try and do is we want to use modular components, particularly cellular therapies, to address these 3 greatest complications: relapse, viral infections, and graft versus host disease. Today I'm going to talk more about virus-specific T cells. So virus-specific T cells, as I mentioned, are a complication. Viruses are a complication after transplant, most commonly from viruses like CMV, EBV, and adenovirus. There are antiviral drugs that treat these, that's well known, but they're not always effective. For instance, cidofovir for adenovirus. They can be expensive, and they can be myelosuppressive. So if you're trying to give a patient a bone marrow transplant and reconstitute their immune system, the last thing you want to do is suppress it. And so giving these antiviral drugs actually can be myelosuppressive, which causes additional viral complications. Lastly, they do have unwanted side effects and toxicities. So you can read all these things in the literature and the infection rate or the disease rate and things like that, but what really resonates for me is that we get 2 to 3 inquiries per week of patients throughout the country from physicians who have a patient that have refractory viral infection and don't have any other alternatives. So to me, that speaks to the need that we have to develop alternative antiviral drugs. So there are other options available. So antiviral T cells have really been deployed for the last 20, 25 years. They used to take 3 months to generate these cells. It was pioneered by Stan Riddell at the Fred Hutch, Rick O'Reilly at MSK, and Cleo Rooney, Helen Heslop, and Malcolm Brenner, then at St. Jude, then at Baylor College of Medicine. Another option is donor lymphocyte infusion, which is just taking non-specific T cells and delivering them. This is associated with graft-versus-host disease. But the option that we're really interested in is donor-directed T cells. So you're taking blood from the bone marrow transplant recipient, we're generating T cells from that donor, and then delivering them to the recipient. Recently, another option has come about, and that's third-party off-the-shelf allogeneic T cells, and these are being pursued by us and a number of commercial companies such as Atara and Alavir, which used to be VirusCite, which spun out of Baylor College of Medicine. And that's what's been really interesting for me. So that's one thing that's different in the last 7 years is now we actually have industry partners who are interested in pursuing antiviral T cell therapy. In the past, you used to have to go to these centers of excellence in order to get those T cells. And so here's a list of at least 3 companies, VirusCite, Atara, this is now Alavir, and CellMedica. And you can see that actually Alavir and Atara, I believe, are going for licensure in the next year or two. And this is what I was talking about, donor-directed T cells from peripheral blood. This is how we can generate these cells. This was adopted from Ann Lean and others at Baylor College of Medicine, and it's run by our PI, Dr. Mike Keller. And all we need to do to generate these cells is take about 20 milliliters of peripheral blood, isolate the peripheral blood mononuclear cells, add the cytokines IL-4 and IL-7, along with overlapping peptides of interest. So in our case, we use overlapping peptides targeting CMV, EBV, and adenovirus. And after only 10 to 14 days of these cells cultured in a G-Rex device, we were able to generate over 200 million T cells that target viruses. And this is an interferon gamma ELISpot assay. I'm sure most of the people in the room are familiar with this, but essentially it's an assay that detects T cell activation. So if you were to dump those overlapping peptides onto the T cells and they recognize them, they will secrete interferon gamma, which are detected in the form of spots, and that's what's depicted on the y-axis. And you see here that our products do target CMV, EBV, and adenovirus. And I should point out this is all within one culture. This is sort of the gold standard for what we would want to see when we give one of our virus-specific T cell products. You see here this patient was not responding to rituximab. They have an increase in the viral load, EBV viral load, shown in red here. We delivered T cells, and there was shortly after a decline in the EBV viral load in the peripheral blood of the patient, and this coincided with an increase in the number of EBV-specific T cells, shown there in blue. So in total now at Children's National, our response rate for donor-directed T cells is somewhere around 80%. Most patients treated did have refractory disease with few other options. And now we have a trial open actually that targets not just CMV, EBV, and adenovirus, but also BK virus, HHV6, and parainfluenza. But the limitation of this approach is that it's only available when the donor is seropositive for the virus you're trying to target. And that's because for starters, has memory virus-specific T cells that you can isolate and rapidly expand. But that's different in a naive T cell source like cord blood, where all the T cells are naive and there's no memory T cells to simply expand over the course of 7 days. And in cord blood transplant, the risk of getting a viral infection is actually greater for that same reason. When you give the cord blood graft, there are no virus-specific T cells to offer protection to the recipient. So we said there's clearly a need to try and develop these cord blood-derived virus-specific T cells. And so this is actually my graduate school project. To generate these T cells, we took an adenoviral vector, shown here, and engineered it to express the immunodominant epitope from CMV, PP65. We also used Epstein-Barr virus transformed B-cell lines called EBV LCL. And these LCL are convenient because not only are they antigen-presenting cells, but they can be transduced by that adenoviral vector, and they also express upwards of 11 lytic and latent EBV antigens. So that's a great source of EBV antigens and an APC. So to generate these cells from seropositive donors, this was 13 years ago now. This is before the rapid method. What we would do is we would take that vector that I talked about, you would transduce peripheral blood mononuclear cells at the first stimulation, and then you would transduce the EBV LCL at the second and third. One of the limitations of using EBV LCL is that it takes 2 months to grow these. And so this process would take about 3 months. So we tried to use the same manufacturing process to generate our cord blood virus-specific T cells. What we found was that we were unable to make these T cells. And we thought about it, and it makes sense. We're not using strong antigen-presenting cells. We were just using monocytes. And we weren't using cytokine combinations that would be present during the priming of an immune response. And so we went back to the drawing board and said, how can we better mimic an in vivo priming condition? So instead of using cord blood mononuclear cells, we use cord blood dendritic cells. We also use the cytokines IL-7, IL-12, and IL-15 at initiation to, again, better mimic that priming condition. What we found is that we were able to expand adenovirus, CMV, and EBV-specific T cells from the majority of cord blood T cell lines that we had attempted. One of the questions we asked was, well, are we actually generating it from the naive population, or are they coming from the memory population? Because in the past, all the T cells that we had generated had come from the memory population. So we sorted naive and memory and then generated the cells just as we had before. And what we found was that the productive T cells were actually derived from the naive population and not the memory population. And again, this is different from what you would find from healthy seropositive adult donors. Another question that we had was, are we actually expanding the cells from the mother? Are these contaminating maternal cells that are expanding? And that's why we're able to generate these. Because again, before this, no one had really been able to do this in a way that was GMP compliant. And so what we did was we took a male neonate and we expanded cord blood T cells from that neonate. We then used fluorescence in situ hybridization to stain for the X and Y chromosome. And you can see here, by the presence of the Y chromosome in green, that this was indeed derived from the male neonate and not from the contaminating maternal cells. And this is perhaps the most interesting part of this entire story. CMV T cells have been around for a long time now, 25, 30 years, and they've been extremely well characterized. So if you are an HLA-A2 CMV seropositive donor, the chances are you have CMV T cells that recognize, for instance, the peptide NLV. If you are B7 and A2, perhaps you recognize TPR or RPH instead of NLV. This is a sort of in-depth characterization we have for almost all alleles. But we don't know what the response from cord blood is. And so what we did was we used overlapping peptide pools to determine what the peptide response is from these T cells. And so what we have here in red is an HLA-A2 CMV seropositive donor. And not surprisingly, this donor recognizes pool 2 and pool 21, which I can tell you corresponds to the immunodominant epitope NLV. However, when we look at an HLA-A2 cord blood unit, which is shown in yellow, instead they recognize pools 10 and pool 17, which corresponds to this pentamer, with this peptide MLN, which we have indeed confirmed by pentamer. And so in over 20 cord blood lines now, we have not been able to generate immunodominant epitopes from the cord blood, whereas every time we do it from a seropositive donor, we are able to. And so far, this seems to be unique to CMV. So in summary, the first part, we can generate these T cells. They seem to be derived from the naive T cells. They're functional, and they recognize atypical epitopes of CMV. But the real reason we did this was because we wanted to see if they could help patients. And so we went ahead and translated this into a clinical trial. So the clinical trial design of this was it was open to any recipient of a cord blood transplant, but it had to have a fractionated cord blood. So for those who aren't familiar with cord blood transplant, it's about 100 milliliters of blood, and this is the 20% fraction. So it can come in a fractionated unit. 80% would go to the patient to reconstitute their immune system, and then this is the 20% that we would use to generate all of those T cells. So it's about 5 milliliters, so it's not 20%, excuse me. It's not 100 milliliters, but it's 5 milliliters. It's about the size of your thumb. And so you can imagine how challenging it is to meet a clinical dose with only this many cells. The cord blood unit must have 25 million cells TNC per kilogram, and this is a phase 1 dose escalation study ranging from 5 million cells to 25 million cells per meter squared. And the cells can be given any day after day 30, which we typically would expect the recipient to have engrafted by then. So before I go further, I do want to clarify that this gets a little bit complicated because we started the trial at Baylor College of Medicine. We treated 9 patients, and then we moved to Children's National, where we opened up to ACTCAT-2, which was the I guess, daughter protocol or son protocol of the initial one called ACTCAT2 at Children's. Very similar design. The only difference was that instead of using that adenoviral vector that I had told you about, we switched to using overlapping peptides because it was easier, and we didn't want to use live virus, so we didn't have to. So we had currently enrolled 12 patients at Baylor. 9 of them received T cells at a median of 92 days, which, that's one of the limitations of the study is 92 days after transplant. We want to get the cells much sooner than that. 1 patient did not receive cells, and 2 patients were ineligible. These are just the characteristics. I know it's a lot. What I want you to take away is that these are malignant and non-malignant diseases, and everyone that received a cord blood T cell line or cord blood unit was a 5 out of 6 or a 6 out of 6 match, and they were all on some sort of GVHD prophylaxis at the time of T cell infusion. Now, one of the limitations of cord blood transplant is that there's a finite number of CD34 cells in the cord blood unit, and you can't get any more. So if you're doing a bone marrow transplant, the physician will write a prescription saying, I want 5 million cells per kilogram, CD34 cells per kilogram, and that's what you go for when you do the bone marrow harvest. With a cord blood, you have what's in the bank. You can't change it. And so one of the limitations is that there's delayed engraftment in cord blood transplant because there's fewer, typically, CD34 cells in the graft. So if we were taking 20% of that already limited CD34 dose, are we going to further delay engraftment? And the answer seems to be no. So if you compare the 80%, those receiving the 80% fraction shown in blue, they seem to actually engraft faster than those who received 100% of the cord blood unit shown in black. I think this is probably not because we're taking 80%. It's probably because in order to be on our study, they had to have a very high cell dose. That's probably why there's a more rapid engraftment. There's been no toxicity to date and no graft versus host disease associated with the infusion of T cells. And when we look at the expanded T cells, we are able to meet clinical dose in almost every cell line that we generate. So shown on the left here, this is ACTCAT. So these were the cells that were made in Baylor using the vector. And you can see an improvement when we switch to the pep mixes here. When we look at the specificity of these T cell lines, you'll see that majority of the lines do recognize more than one virus. So you have CMV here, EBV, and then adenovirus shown here. When we look at the expanded cells, they are a mixture of both CD4 and CD8, and also central memory and effector memory. And in immunotherapy, that's the type of memory cell that you want. And once again, it's a similar slide to what I showed you before. We had a patient that had an increase in CMV viral load shown here in green, and shortly after the infusion of virus-specific T cells, there is a decline in viral load, and we're able to detect these CMV-specific T cells in the peripheral blood of the patient. Now, this protocol was open to patients as treatment or prophylaxis, and so we analyzed the virus-specific T cell response or virus-specific immune reconstitution in the patients that were treated. And not surprisingly, the patients that were treated as treatment had more detectable virus-specific T cells than those that treated or given cells as prophylaxis. And this is what we would expect. In our experience, if there's antigen around because of viral infection, these cells are more likely to proliferate, more likely to persist because they're seeing antigen. Now, it's not 1993 anymore. We can't gene mark T cells just to track them in vivo in patients, unfortunately. And so the best we can do is to use T cell receptor sequencing, and that's what we've done in this study. You can see here, this is basically a map of how polyclonal these T cells are, or the TCR alpha beta usage, V beta usage. What I really wanted to show is here on the bottom, if you look at the reactivation versus non-reactivation, there does seem to be a slight increase in the unique T cell receptor clones in the reactivated patients versus the non-reactivated patients. These are very small numbers, so it's more of a trend. But as I mentioned, one of the limitations of this approach is that we didn't give these cells to the patient until day 93. When they really need them is around day 30. And so because it takes so long to make these cells, they're not getting as much benefit as they potentially could. We had 2 out of 20 lines that didn't meet dose level, and it's really challenging to process these cells because there's a lot of nucleated red blood cells in the cord blood unit. As I mentioned, the median time was 92 days, 93 days, which was too late. And currently we require the use of EVV LCL, which is a live virus and not looked upon favorably by FDA and certainly not EMA. And so this was the old way of manufacturing our cells that we've just gone through, right? We have the dendritic cells that are pulsed with the peptides. After 9 days, they're stimulated with LCL that have been pulsed with those peptides. But again, it takes 2 months to generate these EBV LCL. So the minimum is probably 60 days to make the cells. Using our new method, what we've done is we use activated T cells or PHA blasts as antigen-presenting cells in the presence of these genetically modified K562 cells. So these K562 cells, what they do is they express costimulatory molecules, and so when you combine them with PHA blasts, which are presenting the antigen via HLA, we get basically sufficient antigen presentation. And this is just preclinical data demonstrating that we are able to expand these cells, and they are as good, if not better, than when we use the old way, which is the LCL way. So we do have specificity of these lines. We also now target BK virus because we are told by the clinicians that BK virus is becoming more and more of a problem in transplant recipients. So we now have an open clinical trial called CHEERS, and we've actually treated our first patient now on CHEERS, and this is using that new manufacturing method. If you just take it back a second and look at the cord blood manufacturing compared to the peripheral blood manufacturing, we're still not there. The rapid peripheral blood method takes 10 days, whereas the new cord blood method takes 30. If you look at the cost, it's slightly more, but the specificity and the expansion is just not nearly as good as the peripheral blood T cells, and so that's something we need to work out. Now, I mentioned that probably the coolest part of that project was that we were able to detect these atypical epitopes of CMV. And so we want to dig in and investigate why we recognize those atypical epitopes. And so to do that, we said, well, are there other sources of naive T cells that can target CMV? And the answer is yes. So about 40 to 50% of the population is CMV seronegative. So they should have naive T cells or they don't have a memory T cell response to CMV. And so what we did was we found these donors and we tried to expand CMV-specific T cells using that same methodology that I told you about before. What we found was that, we did a great job of expanding adenovirus and EBV-specific T cells, but we didn't generate CMV-specific T cells. And it all goes back to that same story. There's probably adenovirus and EBV-specific T cells that are memory, and so they will outcompete the naive T cells that are trying to expand against CMV. So we went back to the drawing board and said, okay, at the initiation, let's only use the peptides for CMV, and then at subsequent expansions, we'll use the EBV LCL. And what we found was that we were able to generate CMV-specific T cells from the naive T cell population, and that took some time to optimize. Once again, these T cells were effector and effector memory. And importantly, they were polyfunctional because there's been a number of reports now that demonstrate interferon gamma itself is not sufficient to predict an effective T cell response. Indeed, you need interferon gamma, TNF alpha, other cytokines and activation markers. So once again, we did that epitope mapping using overlapping peptide pools. These are all CMV seropositive donors, and not surprisingly, we see pools 2 and 21, which correspond to NLV. But when we look at the CMV seronegative donors, we don't see NLV. Instead, we see these other epitopes that we've further characterized. So 7 out of 7 CMV seronegative T cell lines recognize atypical epitopes of CMV. Now it's 30 out of 30 cord blood lines recognize atypical epitopes, and 11 out of 11 CMV seropositive lines recognize the typical epitopes. And we did confirm one of these epitopes. This is a novel epitope called LQT. So why do these T cells recognize atypical epitopes? Well, so we came up with a number of hypotheses. Maybe there's an in vitro artifact. So this isn't a real-life antigen presentation. The CMV is not infecting a cell in vivo. That's then, the antigen presentation is modulated and things like that. We're using dendritic cells, so maybe we're not using the appropriate cell type. Maybe there's not correct antigen presentation or cross-presentations, things like that. And then maybe there's a difference in T cell avidity. Maybe there's a difference in the T cell precursor frequency, because we're only looking at a snapshot in CMV seropositive donors that's years down the line. We're not looking at what that response looks like originally. And so to test whether we're using the wrong types of APCs, what we did was we took fibroblasts, which can be infected by CMV, and we cultured them with dendritic cells so that there could be cross-presentation if necessary. And we expanded T cells to look for the typical epitopes. We expected that, we hypothesized that we would restore the typical epitopes by doing this. And this is just to show you that from CMV seronegative donors, from cord blood, and from CMV seropositive donors, we can generate an effective CMV response using the infected fibroblasts. And by the way, this was actually how Stan Riddell did it 25 years ago. What we found was that we were able to detect the typical epitopes in the CMV seropositive donor. So you'll see NLV, TPR, and RPH. So this must be a A2 and B7 allele. But when we look at the CMV seronegative or the cord blood, we do not detect NLV, QID, or any of the typical epitopes that we would expect. Instead, we do recognize that atypical epitope LQT that I mentioned before. So maybe there's a difference in the T cell avidity. So what we did was, first was we generated those same T cells just for the atypical epitopes, in this case MLN, from a CMV seronegative donor, and we see that the EC50 is about 0.8 nanomolar. When we look at that same line from a CMV seropositive donor, it's about 4 nanomolar, so less avid, which indicates that maybe there's some sort of selection of the less avid T cells if for the atypical epitopes. And this is a list of the polyclonality of the cells. You can see that NLV is much more oligoclonal than CMV seronegative donors for MLN, which are more polyclonal. So one experiment we did, we said maybe there's a difference in the precursor frequency of the naive T cells. So we only know about NLV, for instance, because that's what we detect years after the infection, the primary infection. But what if the starting population is actually much different than the ending population, or what we're used to? And so we took 5 cord blood units that were HLA-A2, and we queried those 5 cord blood units for all the T cell receptors that are known for CMV. And actually what we found was that there were more T cell receptors from the atypical epitopes, shown here, than for NLV, indicating that that could be a reason why they're recognizing atypical epitopes, is just because there's more T cell receptors that recognize them. So in summary of the virus T cells at Children's, we treated 80 patients with virus T cells. Between 70 and 80% of them have an overall response rate, depending on whether it's donor-directed or off the shelf. We've initiated that CHEERS trial targeting CMV, EBV, adenovirus, and now BK virus. We've moved to lead 2 multi-center nationwide clinical trials using third-party EBV T cells as well as third-party VSTs. And that's what I'm showing here, is that if you are a pediatric center in the United States, we are running a clinical trial through the Pediatric Blood and Marrow Transplant Consortium, funded by CIRM in California, to deliver these CMV-specific T cells pre or post-transplant. And we've treated a number of patients already in this study. So final thoughts. What is the role of off-the-shelf virus-specific T cells versus cord blood T cells? Obviously, it's much easier to deploy allogeneic third-party T cells because there's a bank of them frozen in my freezer right now, and all I need to know is the HLA of the recipient, and we can immediately deploy those T cells from our bank within 48 hours. Cord blood T cells still take 30 days, and there's still logistical challenges to manufacture those. They're very challenging to make. What will the cost of an approved drug be? You know, it costs us about $8,000 for each product, but what about for an approved drug? Obviously, we all know that in the US, Kymriah costs $475,000, and Yescarta is slightly cheaper. What about T cells from cord blood? So I didn't talk about our work with tumor-associated antigen T cells. We had a paper come out in JCO a few weeks ago for solid tumors that was really promising for us, but maybe we can make those T cells from cord blood. And how can we manufacture enough virus-specific T cell products in a cost-effective way? So with that, I'd like to thank everybody who's been involved. So Kath Bollard is our center director, and she's been a longtime mentor of mine. Mike Keller runs the virus-specific T cell protocol at Children's. Dr. Dave has helped with some of the cord blood T cell development work. Famita Hawk is our regulatory director. And then this is most of our team in the cell therapy laboratory. Alistair Abraham is now the PI of CHEERS and the first author of that Blood Advances paper that I cited. And I just want to thank the rest of my team for all of the hard work and our collaborators. So thank you.
think, are there any questions for Patrick?
Terrific talk. I have a question about the subdominant epitopes.
Yep.
That'll work. Do it over there. Are you convinced that they're naturally processed? Are you convinced that they are HLA-A2 restricted?
Yeah, so they're not, so the ones that we've identified, one of them is A2 restricted, one of them is B35 restricted. So we've identified a few of them. As to whether they're naturally processed, that was why we did the dendritic cell experiment. That was as close as we could get to trying to do natural processing, because we do infect the dendritic cells with CMV using a CMV, a DC tropic CMV strain. And then we let the DCs present the CMV antigens. But yeah, that was absolutely what we were concerned about.
And another comment, another workaround to your dendritic cell problem is to use CD40 activated B cells.
Mm-hmm.
And you can deliver that via K562 expressing CD40 ligand,
No,
essentially. So it's another sort of workaround, especially if you have issues with the GM K562 that you're using.
Yeah, no, that's good to know, because there's obviously concerns about the K562 if we don't irradiate them properly, delivering them clinically.
Use microphone or yell. Great talk. So I'm curious actually about your choice of the K562 as your expander line, which is a much more typical sort of NK cell focused line. Do you think NK cells are involved, or maybe just a little bit more flavor on why you selected that line as your expansion?
Yeah, so we did with and without K562 cells. You know, the PHA blasts theoretically could be enough to expand the T cells after one stimulation, but we found that using the genetically modified K562 did end up with better expansion and better specificity. As to why we use that, you know, we're still searching for an artificial antigen presenting cell that's relatively inert that we can just use, and we don't have to worry about sort of getting rid of it, if you will. But you know, that was what was available at the time. It probably did come from our NK cell work, to be honest with you. We have also generated NK cells from cord blood, and we had them available. We do tend to generate more NK cells when using the K562 cells, likely because that has IL-15 expressed on the K562 cell.
Right. So have you looked at the percentage of NK cells at the end of your culture?
Yeah, I don't know it off the top of my head, but
Okay.
it's definitely appreciably more than when we use the K562 cells. In cord blood and every other we have a couple other different manufacturing paradigms that we use it for, HIV T cells and things like that. And you know, it's 2 or 3 out of 10 will have 20% NK cells or something like that.
Do you think they're involved in your responses?
Yeah, that's a good question. You know, a lot of the literature suggests that cryopreserving NK cells is not great, and so if we were able to get NK cells that were effective post thawing, I would be shocked, but it could be.
So Patrick, just a quick question about the peptide pools. Are they covering all the proteins, or are there some proteins that they're not including as well?
You guys made them, right?
I remember.
I believe what you do is you take the entire PP65 protein, and we did it for IE1 as well, and yeah, you just chop it up into, these are 20 amino acid proteins and then overlapping by 15.
Yeah.
So it does span the entire protein.
I'm just thinking about other proteins as well as PP65.
Right, yeah, so we tried IE1. At the time we were having difficulty generating IE1 specific T cells, and we were trying to get the clinical trial started, so I think we stopped pursuing it. But yeah, this is largely one unanswered question, is it only CMV PP65? I would not be shocked if it happens in other peptides as well, proteins.
And another question was just in relation to the interesting atypical epitopes, which I wasn't aware of that.
Mm-hmm.
So are you also, have you kind of looked at those atypical epitopes in other populations? I'm not just cord blood or peripheral PBMCs, but also other compartments?
So our tumor-specific T cells, we're doing a lot of work now to epitope map the responding T cell, the responding epitopes, but we just don't know what the memory response is versus the naive response, and so we don't know whether they're atypical or typical yet.
And have you seen those MLN epitopes in anywhere else apart from cord blood cells?
Oh yeah, so the CMV seronegatives recognize those, and actually, yeah, we can generate CMV seropositive derived responses against the atypical epitopes. They just seem to be less avid, which is really curious, as if the immune system doesn't want you to develop effective, or the virus doesn't want you to develop effective responses against it. Yeah.
So in the 20% non-responders, what do you think is the reason for non-response in those cases, or the distribution, I suppose?
Yeah, in a GMP facility, you should have everything standardized, everything should be the same, but in the reality, people are different. And so the first line that we made using a different technician did not work. So I don't know if that was training or that was just a bad line. Generally, when we don't get productive responses, the number of mononuclear cells we get from the cord blood is very low, like 40 million cells. And to make dendritic cells plus B cells or LCL from 40 million cells is difficult.
And so if you sort of compare your current method to multiple selection, where would you sort of say that the relative technologies are in your mind?
Yeah, so I think I had a slide on that. The limitation from cord blood is that if you're using naive T cells, the selection via multimers is going to be really challenging because the frequency is so low. I do think it's an effective method. I got a call from Boston. We're looking to build our VST program. We don't really have a lot of resources. We just want it as a therapy for our patients. Do you recommend using multimer selection, or do you recommend using your rapid VST program? And if you're not really willing to do a lot of the characterization, and if there's not a research project behind it, I would recommend just using the multimer selection because it's easy. If you can buy a Prodigy for a lot of money and just select a product and give it to the patient. So, yeah, I think there is merit to that. It can be expensive. You need to do an apheresis. We can use 30 milliliters of blood. So there's trade-offs there. But, if you just want protection for your patient, I think that these two are suitable methods to do that.
So I have a question about the atypical epitope also.
Mm-hmm.
So I'm not sure if I understood. So when you generate these responses, you're using adenovirus that is just expressing the MP6. So it's not really seeing the virus as a whole for CMV, correct?
Yeah, so we've generated the virus-specific T cells a number of ways. We started with the adenoviral vector that has a transgene for CMV PP65. Absolutely right. We then moved to just using overlapping peptides. And then in one of the experiments, I showed that we can actually take a CMV that can infect dendritic cells and generate them that way.
And
we still see the atypical.
So it's not like there is an immune evasion
Yeah.
for this virus protein that is inhibiting the production of these.
Yeah, with the caveat being it's still in vitro. It might be infecting a different cell type in a different location in the body. There's a lot of factors that are going to be different. Yeah.
And do you know if this atypical epitope inhibits viremia?
So we did an experiment where we generated our atypical specific T cells, and then we infected fibroblasts with CMV. We co-cultured the T cells that we made with those infected fibroblasts, and then after a few days, we took the new CMV and re-inoculated new fibroblasts, and both the typical and atypical were both capable of preventing dissemination of the virus.
Thank you.
Yeah, thank you.